Welcome. Welcome, everyone. Good to see you. Before we get started, this would be a nice little gentle reminder to turn your cell phones to silent. In a group this size, I'm not sure if you want to be that person. Um, oh. Anyway, so in case you can't tell, you are at Breast Cancer and Beyond Advances in Women's Health from Biomedical Engineering at Tufts University with Shelley Peyton, Professor and Department Chair of Biomedical Engineering. This is our final lecture in our Faculty Spotlight series. And so my name is Marianne Hunt, and I am the Director of Lifelong Learning in the Office of Alumni Engagement. And we do these programs specifically because we know the alumni are always intellectually curious. They're always interested in the exciting research and innovations that are coming out of Tufts. So it really is a pleasure in my job to get to know faculty across all of the schools and to hear about really super exciting research. Sometimes I feel like I'm like the zealot in the room whenever somebody asks me about what's going on at Tufts, and they can't actually shut me up sometimes. um, so yeah, that's what I was going to say about the program. But with that, I'm just going to get to introducing Shelley. She has a very wonderful and a lot of high credentials in her bio. So, and excuse me for reading because this is my third third faculty I've introduced this weekend. So Shelley Peyton is a professor and department chair of biomedical engineering, as I already said. She received her Bachelor of Science in Chemical Engineering from Northwestern University and went on to obtain her master's and PhD in chemical engineering from the University of California at Irvine. She was then an NIH Kirstein postdoctoral fellow in the biomedical engineering department at MIT before starting her academic appointment at the University of Massachusetts Amherst, and then finally coming to Tufts in 2024. She leads an interdisciplinary group of engineers and molecular cell biologists seeking to create and apply novel biomaterial platforms towards new solutions to grand challenges in human health. Her lab's unique approach is using their engineering expertise to build simplified models of human tissue with synthetic biomaterials. I don't want to spoil it for you, but they use these systems to understand the physical relationship between metastatic breast cancer cells and the tissues to which they spread, and also the role of the extracellular matrix and its dynamics in drug resistance, and also how to create bio-inspired, mechanically dynamic, and activatable biomaterials. Among other honors for her work, Shelley was a 2013 Pew Biomedical Scholar, received a New Innovator Award from the NIH, and was awarded a career grant from the NSF. She's a fellow of the Biomedical Engineering she's a Society and a fellow of the American Institute for Medical and Biological Engineering. She is passionate about graduate student training and diversifying the academy. She was awarded an Outstanding Teaching Award from the College of Engineering at UMass in 2018 and has led an REU site co-directed a biotechnology NIH training program and was the lead principal investigator of a prep program at UMass, which hosts students from historically excluded groups for a one-year research intensive program to help them prepare for graduate school. So with that, please help me welcome Shelley Peyton.
Thanks, Marianne. Can folks hear me okay? And it sounds like the microphone's on too. So I'm going to set a timer. Marianne said I had 30 minutes, and faculty will drown you out for as long as we possibly can until you rip the microphone away. So that'll keep me on time. And we have a fairly small group, so I would totally welcome interrupting me, asking questions as we go. Don't feel the need like this is a movie or a Broadway show that you have to wait for questions till the end. Okay, so Marianne gave a little bit of my background. This is actually where I'm from Coffeyville, Kansas. Has anyone been to Coffeyville? How about Kansas? Yes, a few from Kansas. So if you ever need a drive from Tulsa to Kansas City, you'll go through historic Coffeyville. So that's where my mom's side of the family is. We moved around a lot as a kid. Kansas, Illinois, Wisconsin, Kentucky, Minnesota, always in really small towns. And that's a little bit about how I ended up at Northwestern. I just wanted to be in some city. where I could also get a fantastic education. And I think Tufts is a lot like that too. So back when I was an undergraduate at Northwestern, this is when the Human Genome Project was really coming out. And that's where I got my start as a researcher, as an undergraduate in the lab, working with Annelies Barron to basically do chemistry on very small capillaries so that you can get DNA to flow through really rapidly so that we could sequence it better. As Marianne said, I then went on to study tissue engineering and cardiovascular disease. At UC Irvine, we were using this concept of building tissues, not as something that would replace an organ in the human body, but really to use them as better model systems so that we could study human disease in a better way than what we can achieve with animal models. So we were trying to understand how stiffening of arteries, which all happens as we age, how that can trigger heart attacks and plaque buildup. I then went on to finally move to Massachusetts. I think of myself as a true New Englander now. I've been here longer than I've been anywhere else. I went to study how cells move in three-dimensional environments at MIT. And that's kind of how I got my start as a researcher at UMass Amherst, because when cells move when they're not supposed to, that can be the start of a really bad disease. So this is how I bridge these interests from building model tissues to cell movement to think about breast cancer metastasize and why does it move to places that it shouldn't. And once it gets there, how do we kill those cells? Tufts has an amazing program, both on the Medford campus, but also at the med school, studying breast cancer and many other cancers. And so I was so elated to be recruited here almost 2 years ago, where we're really taking our research in brand new directions because of Tufts support. Okay. Any biomedical engineers in the room? Engineers in the room? I get a lot of questions about what is really biomedical engineering. I just came from the senior lunch before coming here, where we're celebrating all of our biomedical engineering seniors, and they fall into all of these buckets, right? Trying to think about using engineering fundamentals like understanding transport and understanding circuitry, understanding thermodynamics, and then applying them to solve grand challenges in human health. And you don't need a biomedical engineering undergraduate degree to work as a biomedical engineer. It's really that application to human health that makes anyone a biomedical engineer. Okay, this is what I study. So the statistics that I'm showing you here are for the US and Europe, Western world. These statistics are a little bit different when we look at populations in the African continent or Asia. Okay, so just keep that in mind when I'm showing you these statistics. But in the US and Europe, right now, about 1 out of every 7 women will be diagnosed with some type of breast cancer in their lifetime. Okay, what is breast cancer? It is this disease of, like other cancers, uncontrolled cell growth. So every time your cell has to divide in your body, which is happening all the time, it's got to copy all of the elements inside, and that includes the DNA. Cells are very, very good at that, but every once in a while they'll make a mistake when they're at the photocopier. It's very, very rare that they make mistakes. Your cells actually have wonderful machinery to, when there's a little kink in the chain, they can come back and repair it or splice that out. But we still pick up these mistakes over time, okay? Hopefully that mistake is made and a cell catches it, and what it will do is we'll go through programmed cell death. They're like, there's an error here, I should not live anymore, and it will go through a programmed cell suicide. Or you can have mistakes in DNA that are just in non-coding regions. They're in non-functional regions of the DNA, and so it doesn't matter. They can live there forever and not cause any problems. But every once in a while, a mistake will be made in DNA that codes for a gene that regulates cell growth. So now that cell that has that mistake is dividing faster, and it's passing on that mistake to all of its progeny. Because those cells are dividing faster, they're making more DNA and they're making more mistakes, right? And so that's how you end up with this disease of uncontrolled cell growth. If you can catch breast cancer early, it is incredibly survivable. So stage 1 breast cancer, the survival rates in the US and Europe are 95 to 99%. And that's really due to wonderful technologies that have come out in advanced imaging and early diagnosis, right? That has really revolutionized breast cancer here in the US and Europe since the 1970s. And you can tend to get better treatments, right, that are less toxic and have less side effects if that breast cancer can be taught early. So one of the advocacy things I do in my classroom is just encourage everybody in the room to do self-exams, right? Every month, make sure you're doing that. If you can catch it early, very, very survivable. Unfortunately, there's a portion of women who catch that disease late, or a very, very small percentage where it can spread, even if that early diagnosis. And once that disease becomes metastatic, some oncologists still call this disease untreatable because now the 5-year survival rates drop to 30%. So the huge difference, right? And so why is that? Well, it's very hard to surgically resect tumors that are in the brain. It's hard to survive without regions of your bone marrow because that's responsible for making all of your immune system, right? And so it ends up in these places that we just can't live without, whereas we can get lumpectomies and mastectomies, right? Although painful, very survivable. Also, for reasons we still do not understand, breast cancer cells that are in the skeleton, for instance, or in the lung, become really drug resistant. So those drugs that were working to kill cells in the breast environment no longer work when they're in the bones or the liver or the lungs. And so this is why my lab, although this is a very important problem to solve, we're really working here to help those patients that have these 30% survival rates, right? And that's also, we had one, Marianne, you warned them. This is really the rate limiting step, is the ability of cells that have left that original tumor to survive and grow at these secondary sites. So what happens during metastasis, how do cells get there? There's a primary tumor in the breast, okay? Cells need to detach, right? They let go of their neighbors. They need to find a blood vessel or a lymph vessel nearby. They then need to get in to that vessel. That's also a torturous process because you can imagine this cell being just thrown around throughout the circulation system. Many cells die throughout all of these steps. They then need to get to a secondary place and get out of that blood vessel. And then they need to turn growth back on. So there's many, many, many different steps that a cell has to go through, which is why it's thankfully a rare event, right? But it's also the most powerful cells and the most drug-resistant cells that can survive that process and then grow at secondary sites. Okay, one of the most confounding factors about metastatic spread, and you can look across different cancers, we've known since the late 19th century that metastasis isn't random. Breast cancer cells don't go everywhere, right? They go to these very specific locations. Prostate cancer is another really challenging disease to understand because for reasons we don't understand, when it metastasizes, it only goes to the bone. You very rarely will find these prostate cancers metastasizing other places. And you can see some of these other patterns of spread that emerge. And so I think rightfully so, cell biologists, people that really think about genes and DNA have thought, well, there must be some genetic driver. What's different genetically about a prostate cancer cell versus a stomach cancer cell that allows it to have this what's called tissue tropism, this ability for cells to end up in specific places. But even though we've been trying that technique as a field since about the 70s, we still don't know how this works. Within breast, there's also this challenging aspect of knowing where a patient will have metastasis. And there's some link between where cancer cells will go and what the subtype of breast cancer is that a woman is diagnosed with, right? So let me give you a few examples, okay? So luminal A is one of these subtypes of breast cancer that a woman can be diagnosed with. And the way that a pathologist does this is they look at hormone markers for cells on the tumor, and they're saying, well, how many of these cells are estrogen receptor positive? How many of them are HER2 positive? How many of them are progesterone positive? And that allows them to bin the cancer into these different subtypes, and that can really inform treatment because you might be able to target the estrogen receptor, right? So luminal A is the most survivable statistically of breast cancers. It expresses all of these hormone receptors in normal amounts. That means we can target it with things like tamoxifen and Herceptin, okay? And for reasons we don't understand, it actually doesn't metastasize very frequently, okay? When it does metastasize, it almost exclusively goes to the bones. That's where that's found in women. HER2 overexpressing breast cancer means exactly what you would think. It makes a lot of this HER2 receptor. It means you can target it with Herceptin, but it tends to be drug resistant. It's highly metastatic, and these women tend to have metastases to the bones and the lung and sometimes to the brain. And then triple negative breast cancer is negative for all of those receptor markers. It doesn't express these hormone markers, meaning we can't target it with those hormone targeting drugs, which means women are getting chemotherapy, right? Very powerful side effects. And it's highly metastatic, and you can find these triple negative breast cancers everywhere throughout the human body. But again, even though we know about the pattern, there's no known relationship between estrogen and bone metastasis versus lung metastasis. There's not a concrete link there, right? And we know it's not driven by at least genetics alone. So this is why we're trying to use tissue engineering to understand the relationship between these disseminated seeds coming from the tumor and the soils in which they land. We're building the soils essentially to study why do patient cells grow in some soils and not others. And once they're there, the most important thing is then how do we devise a therapeutic regimen that's gentle but will kill those cells that have invaded that foreign site. I would welcome any questions so far that have come up. Yes.
Examining the soils to
Yes.
see where they land, do you think there's also something about the different cells from these different types of cancer that has to do with why they choose to go by the bone or whether to go into the bone?
Yeah.
Does it originate with the soil or does it start with the nasty seed?
It's a great question. So what we're discovering as a field is that these breast cancer cells actually get everywhere. So people can find breast cancer cells in skeletal muscle, can find them in the spleen, okay? There was a study that came out about 15 years ago where they looked at bone marrow from all stage 1 cancer patients that had then passed of something else, and they found breast cancer cells in the bone marrow of all of those patients. So now the prevailing thought is that cells are actually good at getting everywhere, but for reasons we don't understand, they're either growing at those distant sites or they're dying or sitting there dormant throughout a person's lifetime. So that's why we focus on the soil and what might be different about different patients' soils, so to speak, what might be different about lifestyles, and what might be different about these surrounding tissues. Other questions? Okay. So to a cancer cell, these mimics of tissue that we're building can trick them into thinking that they're in the bone marrow or they're in the brain because we're providing chemical and physical signals that mimic those tissue sites. To you and me, it looks like a small pile of snot. Okay, so we've engineered these in a way that we're not scooping out bone marrow from patients and using those. These are fully engineered from synthetic polymers and parts of proteins so that we can make them very cheaply. We can make them very small. They're optically transparent, so we can see through them, right? Because one of the challenges of finding cells in the bones is we cannot see through the bones, right? Those have to come from a bone marrow aspirate. So what you can do, and I'll play the movie again, it takes about one PhD thesis worth of work to design the bone marrow mimicking tissue or the lung tissue. But once we know how to make them, if you can pipette, you can make these. And we actually share these with a lot of different researchers for all kinds of different applications across the country. And so you basically combine this liquid pre-polymer solution with cells, you pipette up and down, and it snaps together in place. It's like a quick setting Jello, essentially, right? And then this is an image of cells being cultured in one of our bone marrow mimicking environments for 2 weeks. So what you see here in green, those are triple negative breast cancer cell lines that are in these small spheroids that are growing over time in the bone marrow. And then the red, more spindly-like cells, you can see them kind of in the background here. Those are healthy human bone marrow stem cells that you would find in really high abundance in the bone marrow where these breast cancer cells are trying to grow. And so we're using these environments to study how these cells interact as well. And can you find drugs that will kill those triple negative breast cancer cells without killing your healthy stem cells, which is one of the challenges that we're working on. Okay, so I thought I would share with you just a few things that the Tufts graduate students are working on in the lab. So again, we're using all of these synthetic tissue environments to study breast cancer and other things. Tiana is studying the intersection of age, race, and ancestry on breast to bone metastasis. So how does menopause change the bone and bone marrow environment, and how might that stimulate cells to grow late in life? You have a question? Okay. Haley is making new cell-derived matrices, so sort of a counterpart to the synthetic materials that we make. She's taking healthy cells from tissues all over the body. She's got a panel of 25 of these, so spleen cells, brain cells, kidney cells, skin cells, and making matrices for them so that not only do we understand why breast cancer cells do go to certain tissues, why don't they grow in the spleen or the kidney or the heart? Because maybe those negative interactions can really inform us about how we kill the places that they do go. Nanette is studying small cell lung cancer in our lung environments. Juan is studying traumatic brain injury in our brain environments. And we have a few new. Maddie is a new student to the lab who just joined in September. She's studying pediatric gliomas in our brain environments, a very rare but incredibly deadly disease. And then Molly and Emilio are new postdocs that are studying sort of across these boundaries. And one note I'll say is that just the ability to recruit amazing graduate students and postdocs has just shot up 500% since moving to Tufts. People love this place. Even though they didn't get an undergraduate degree here, they love the environment. They love how collegial the researchers are here. They love how easy it is to collaborate across labs. And honestly, they love Boston and doing science in Boston. There's no better place to do biotech, right? Okay, so I thought, let's see how much time I have because I might, 10 minutes, this flies by. So I'm going to skip ahead a little bit. This is a cool video of breast cancer cells moving, sort of pseudo-metastasizing in our lung environment, so you can get a sense of what they do in these optically clear hydrogels. And so we're trying to find conditions that cause the cell in the upper left that die versus the cells down here that are clearly moving and growing, and what are the differences there. And then I'll just skip ahead to tell you what I think is really cool about Tufts, both the School of Engineering and Biomedical Engineering. So Biomedical Engineering is 24 years old at Tufts. Started in 2002. We started with 7 undergrads. And since then, we just graduating 59, right? So you can see the growth there, right? And we guarantee independent research in labs or at the med school or with partners for anybody who wants it. Biomedical engineering has done such a great job of that. The School of Engineering is like, that's a great idea. We're now going to require that every department does that. So within the next 2 years, every department needs to comply. Electrical engineering, computer science, any undergrad that wants to do research in the lab will get that chance. It's really cool. These are the 9 undergraduates that work in my lab, and they are awesome. Okay, start them out a little slow, right? Okay, well, let's make sure you can pipette. Let's make sure you can turn on the $500,000 microscope without breaking it, right? And they get sort of a semester worth of onboarding there. And really then the goal is to develop an independent project for these students, right? They're going to take that project in creative ways that I never would have thought of. And they'll have that on their resume to get whatever job they want after the fact, or go to med school or get their PhD, right? So I thought I would just give you a couple of examples of what people are doing. So this is Harper. Harper is a sophomore, right? So she just finished her sophomore year. She joined the lab as a freshman last year, wanting some research experience. And she is taking these fibroblasts, or specific, it's sort of known as a skin cell, but these are the cells that make the materials of your human body, all the proteins and sugars that are there. And she's taking these cells, she's growing them for several weeks at a time. This is painstaking. And then staining and analyzing the materials that those cells leave behind. And she's trying to understand like why this brain material looks so different from a skin material, and how does that impact metastasis. This is Miru. Miru is also a sophomore. She joined my lab last year. So her and Harper are both hating on physics 2 right now. That's where they are in their curriculum. And Miru is using our brain environments to study traumatic brain injury. Okay. So I've had concussions from sports, right? We can think about athletics in the US. We can think about people that have served in the military that have these concussions. And what she's doing is understanding, well, you might have a minor concussion one day and be able to maybe be a little dizzy, but recover from that. But what happens if you have another fall 2 days later? And how destructive is that, and how much of a risk factor is that for Parkinson's, right? Or early onset dementia. So she is using our brain hydrogels that we work with, and she's culturing brain cells in them. These are astrocytes. And she's essentially taking a needle into that environment, and she's blowing a small bubble. And she's studying the forces of that bubble formation and collapse. And what is a force that an astrocyte can recover from? What is a force that will kill that astrocyte? And what is a force that is gentle enough to keep that astrocyte alive, but it'll die a few days later from a second fall? It's just amazing stuff that a sophomore is doing, a second-year undergraduate. This is Leah. Leah is a junior. She loves physics. She actually won the university-wide physics prize last year, but she's studying lung cancer and using a combination of our tissue engineering and genetic engineering to find new ways to kill drug-resistant lung cancer cells. So these are images that Leah has taken on this fancy microscope, where she has, you can't really see in gray, those are the wild type lung cancer cells. In red here, these are drug-resistant lung cancer cells that are often in these tumors at very low amounts, but if you let them grow enough, they'll take over the tumor, and then that lung cancer is no longer treatable. And then in green, what she's done is she's genetically engineered lung cancer cells to also be drug resistant, but then to make chemotherapy. So she's applying drug. You can see the red cells start to grow up a little bit. So do the green cells. And then she turns on a switch, and the green cells make a bunch of chemo and kills the tumor, right? So you can think about this as a localized treatment where the chemotherapy is only going to be in the lung environment. You want to have it systemically because we've tricked the cancer cells into making the chemo for us. So it's a junior undergraduate that's working on this really cool stuff. Okay, so what I thought I would then do is kind of take a pause. That's a snapshot of what my lab does and how Tufts students are really contributing to this research. But we, as a department and at the medical school, are actually quite strong and continuing to grow in our women's health and women's health engineering. And so I just thought I would give you a sense of what other people in the department are doing. Madeline Oden, Theo Omenetto, and Juan Necco. So this is Madeline's work. She's also studying triple negative breast cancer. She's actually one of the big reasons that I wanted to come to Tufts was because of the opportunity to work with Madeline. She's actually trained as a cell biologist, and so teaches a lot of our biology courses for the biomedical engineers, but then works in an engineering department. It's really important, these partnerships that we're building across disciplines, right? So she's trying to understand how these cells spread with a specific focus on the materials around the triple negative breast cancer cells that lead to metastasis, as well as nerves. Yes, so triple negative breast cancer means that that cancer cell does not express the hormone receptors that we would maybe target with drug. So estrogen receptor, progesterone receptor, and HER2. If it's triple negative, it means it's negative for all three, which means we cannot use those hormone targeting drugs to kill those cells. That's why people get chemo when they have triple negative breast cancer. Yeah. And so she's actually studying some also fascinating work about nerves. So peripheral nerves that come from the spinal cord can actually get into a tumor and facilitate metastasis. Cancer cells migrating on these neurons to get to secondary sites. And that's a new area of study that her lab is tackling on how to stop that. These are just some great pictures of her work. Theo Omenetto is trained as an optics engineer and a physicist, okay? And he's building materials that can be used as sensing for the vaginal microbiome, okay? So women can suffer from bacterial vaginosis when certain elements of that ecosystem get out of whack, either typically with respect to pH, but it can be hormone changes as well, right? And it can be very, very difficult to treat, and it tends to be recurrent, right? And what happens, what we know about this disease is that before it gets into actual diagnosis of clinical bacterial vaginosis, there's this transitional state that can be targeted, but it's very, very difficult to know when women are in this transitional state because they don't tend to come into the clinic until it's too late. And so he's creating out of these materials that you can put into underwear liners that have colorimetric sensors that can sense when you have a healthy vaginal microbiome versus bacterial vaginosis, as well as this intermediate state. So you can come into the clinic before you have this full-blown vaginosis. And so he can sense these changes in certain enzymes that are important, hydrogen peroxide, as well as pH. And you can see these color changes. And so the concept here is that a woman can have an app where they're taking a picture that's going to their doctor, and it says, come in, right? This is the time, you're in this intermediate state. We can treat this very, very gently before it gets to vaginas. So a fully different type of engineering, right, from biosensing that's applied to women's health. Okay, and then finally, Juan Necco. He's been at Tufts now for 4 years. Like me, he's a tissue engineer, building tissues, but instead of using that toward cancer, he's building tissues that can help us solve problems in infertility and endometriosis, okay? So he's studying the endometrium. So about 10% of women suffer from endometriosis. We don't think it's that large because a lot of us will suffer from menstruation-related pain and then just deal with it or told to deal with it, and it's no big deal, right? But now we're understanding that this is a real clinical problem, and if left to be too aggressive, it leads to infertility, right? Okay, so he's studying this phases of the menstruation system and modeling that in a fully endometriosis on a chip tissue engineered device, right? So he's obtaining through partnerships at the medical school tissues from patients that they have donated of healthy endometrium, but also pathological tissue from endometriosis, gathering these tissues, looking at what the expression of very specific markers are in those tissues. How does the healthy endometrium really differ from pathology? And then modeling that with these tissue engineered materials. So these are very, very small droplets that are very like the little jellos, the piles of snot that I was showing you earlier, right? But where he's growing these endometrial tissues within, and they look very much like the actual disease. So he's again trying to study what is that transition from healthy to endometriosis. With these endometriosis tissues, we know that some patients respond to treatment, some don't. Because he's got this access to donor tissue from the medical school, he can actually pull from patients that are very willing to donate their tissue to understand why they are or are not responding to treatment so we can overcome this. And these are just some pictures of some of those gels. Okay, I always want to end here with a call to action, okay, about women's health engineering. Please talk to people about women's health, right? I think these are important conversations to have with our friends and family. Advocate. So Marianne, as part of my introduction, said that I was a fellow of AIMBE. This is the American Institute for Medical and Biological Engineering. We have a number of these fellows in our department, and the goal of that society is advocacy. So once a year, we travel to DC, and we have meetings on the Hill all day. And we talk to our representatives about the importance of funding scientific research so that we can develop the next generation's cures, right? These might not be cures for me, but they could be cures for our children, right? It is very easy to go to that website and say, I want to write a letter to my representative. You plug in your zip code. These letters are pre-written for you. They talk about the importance of funding the NIH, of funding the National Science Foundation, and providing opportunities for researchers, right? We've done the work. All you have to do is click and put your zip code and push go. Please encourage more students to come to Tufts. As you know, this place is awesome, right? And I just love working with the talented graduate and undergraduate students here, and they're so passionate about applying these fundamental engineering tools that they're learning to important problems like women's health and many other things as well. And it's pretty easy if you were interested in trying to sponsor an undergraduate student working in the lab, something like a couple of $1,000s to sponsor their work in the summer, right? So I'm happy to answer questions about how you can do that so we can get more of these undergraduates, these research opportunities in biomedical engineering or in the school writ large. That's all I have. Happy to take questions.
Yeah, no, we have time for questions. And I should have mentioned at the beginning that we are live streaming this for a virtual audience. And then we also will post the recording online on our alumni YouTube channel. So feel free to share with friends. But that was my preamble to say, please wait for the microphone for your questions because we want to be able to pick it up for the recording. So there was a question right here already.
So at what point in the maturity of a cell, cancer cell, does it say, I'm out of here? I think I'll find a way out. And does it give a little signal?
Oh, good. Like I'm leaving and here's my goodbye letter kind of thing.
Exactly.
Yeah. So yes. So when cells first leave that tumor, typically they're the recipient of signals. So what happens early on in the disease is the tumor is growing. It needs more oxygen and more nutrients, and it needs it really quickly. And so the first thing it does is these cancer cells actually send out signals to say, give me more blood vessels. Okay. Blood vessels come in, but they're a hack job. Okay. As in they're made really quickly and poorly. Okay. That is actually important to metastasis because these blood vessels come in and they're half full of holes. And that helps these cancer cells get in to those blood vessels and leave. So some of the very early treatments for metastasis, we're trying to say, well, let's stop that. Let's stop blood vessels from coming to the tumor. That should really help. And it actually it didn't, well, it's not a cure for cancer, as we all know, right? But what was the problem was if there's no blood vessels tumors, I also can't get drugs there. Right. So now you have this tumor that's getting fed in other ways and it can co-opt other strategies. And so, yes, so the cell is responding to the sloppy blood vessels that are being made, and that's how it gets in. But it's communicating not necessarily with the cancer cells, but it does communicate with the immune cells in the environment. So once it starts to leave, it talks to the immune cells to say, nothing to see here. Go away. I'm shielded. And those are a lot of the signals that are being studied of how they shroud themselves from the immune system.
Interesting. Other questions? Yep.
I have a question about the system of postdoc. I live down next to University of North Carolina. They have 1,000 postdocs. And I was at an award session recently, 3 foreign students and 3 US born. One is going to the University of Utah to do cancer research, associate professor.
There's a great cancer center there, the Huntsman Cancer Institute, Utah. Yeah.
Now, a year ago, there was a cloud created by Doge and all the other stuff of the new sheriff in town. Has that sort of worked its way out? Do you have people in the pipeline coming who want to be postdocs at Tufts?
Not sure how gently I should answer this question. So I think one of the things that you're alluding to are the travel restrictions and how difficult it is for people from certain countries to obtain visas. That has absolutely affected graduate students and postdocs to come, not just to Tufts, but to the United States. This is an individual decision, but a lot of people also just don't feel safe coming to the US right now. And it's affected. So Nanette that I mentioned, who's working on lung cancer, she's from Burundi. It's a very small country in Africa near Rwanda and Kenya, very poor country. And she is from one of these countries that is affected, and she is not sure if she's going to be able to even get employment here because of the visa issues for new PhDs, or whether she will have to go to Europe to do work or even go home. And that is just devastating, not just for the individual and for Nanette, who really wants to make a difference here, but also for the brain drain that happens in the United States. I think we're missing out on really, really top talent because of these travel and visa restrictions.
Other questions?
What would be the biggest risk factor to getting breast cancer? And is there a relationship between breast cancer and ovarian cancer?
So both of these diseases, breast and ovarian, are hormonally regulated. Ovarian cancer can be much more devastating because it's so asymptomatic. So a woman might have abdominal pain for a while and kind of deal with it, and then finally go into the clinic and it's already stage 4. So ovarian cancer is diagnosed very, very late. And so the treatment options for that are typically platinum-based chemotherapies. It's just a different ball game than breast because of the late treatment, right? Late diagnosis. They're both regulated by hormones. There can be a risk factor. So BRCA1/2 mutations can pose a small additional risk to getting breast cancer or ovarian. And so some women have gotten that genetic identification and have decided to go through a hysterectomy or even a double mastectomy to be preventative. Some women also do this because of the potential hereditary risk if you've got family members. That is actually, we're finding it's less hereditary than we thought. So that's potentially good news if breast cancer or ovarian cancer has run into your family, although BRCA1/2 does still hold a relationship. The biggest thing an individual can do that we found about breast cancer to prevent breast cancer is to have full-term pregnancy before they are either 16 or 18. So that's not great news, right? Because now we're dealing with maybe a public health solution. But yeah, there's really, I wish there was more to say. Breast cancer is one of these cancers that can have mutations in many, many, many different places. And it's sort of the blending of those. There's no one thing that is yes, no, on you will get breast cancer.
Other questions? Oh, okay.
It's a question that I think has been talked about for many years. And at this point, I'm very confused about it. And that is the effect of hormone therapy, including estrogen, as you go through the years, and how I think the latest is it's sort of okay to take if you're just now menopausal, but you shouldn't start it when you're later. And why on earth would that make a difference? And-
we don't know.
Should I even ask that question?
So I think, I guess my answer to this, and I'm not an oncologist, okay, and I'm not an OBGYN, okay, and I know how important HRT can be, perimenopause and menopause. Trust me on that part. I would say if you've had a breast cancer diagnosis and are now in remission, I might personally think about maybe pausing giving like taking more estrogen. But without that, I would say take it, make your symptoms better, right?
Nope. All right. I was actually wondering if we had any doctors in the room who might have something to add to that.
Maybe you have an opinion on this? Okay.
I just was curious about the traumatic brain injury research in terms of, were you researching whether someone who had a traumatic brain injury would be more susceptible to cancers or just to other brain illnesses?
That is a fantastic idea that I do not have funded.
That's not what you're that's what you may.
Well, I would love to do that in the future. So actually, no one's thinking about this, right? So let's say there could be these cancer cells in the brain that are asleep, right? That aren't growing. But if someone would have had a fall, that brain tissue, it doesn't remodel well, but it does try to remodel. These astrocytes in the brain become reactive. They produce a bunch of signals. Are those growth stimulatory signals? But there isn't any clinical evidence about this, I think, because the numbers are so low.
Would other cancers like attached to those spots, or?
We don't know. The question was, what other cancers attach to those spots? Like, could it also be a risk factor for glioma or glioblastoma? We don't know the answer to that.
Other questions? Okay.
First of all, thank you for your talk. And I wanted to tag on to what this gentleman said earlier about student visas and so on. What about the cutbacks in funding for science in general? How has that affected the program here? Or is it too painful to talk about?
No, it's my other hat is I'm department chair, right? So I have to think about this all the time. Yes, it's been very painful. So I can give you some anecdotes about this. We are taking in far fewer PhD students into the department last year and this year because of the federal uncertainty. So there are things like a grant was awarded, but we never see the money, right? Or it's trickling in at really slow amounts. And so we just have to act really conservatively because we don't want to take in a student, even if we think we have the grant for them, because the money's not coming in, right? And Tufts University is working really hard to figure out, like, well, do we pull off the endowment or something to help fund students? But then that puts the university at risk. These are really challenging questions. Yes, we're all sort of diminishing our research by, I would say, on the 15% range, because, I'm thankful that I have amazing colleagues. And so maybe we're not being as affected as some other places. And we're also not currently on the list of universities that the executive office is really harshly targeting, right? That you've probably heard about in the news. So we're good enough that we have amazing people, but we're not in these like top 4 in the news universities where we're really a prime suspect for the executive office to go over. But yes, it's being affected.
We can do genetic testing now to see where there are gene mutations where someone might be a carrier or
Yep.
have a high risk of a particular cancer.
Yes.
Is that a very promising area of research to be able to kind of turn off the mutations or fix those?
Yeah, we don't. So maybe in the future, with advances in genetic engineering, we could think about turning these mutations off or splicing them out. We're not there yet with the BRCA mutations. I think so in certain cancers, there are more definitive driver mutations that really drive the disease. So APC, for example, in colon cancer is a very big driver mutation. It's very predictive of the disease, right? There's the Philadelphia chromosome that we know from leukemia, right? Breast doesn't have this. Okay, so BRCA2 is there. There's P53. There's PLKC, and there's 75 others that could be at these high amounts. So we don't have, unfortunately, this one thing that we could go after genetically, which is why engineers are trying to go at it from these other angles, like the tissue environment instead.
I'm getting my workout in here.
Thank you. And could you just comment on epigenetic engineering?
Yeah, so epigenetics are basically modifications to the DNA that can turn off a gene. Okay, so they put a little methyl tag or something else on there that basically turns off the gene. And that's how your cells in the body do different roles. Not all your cells have the same genes turned on and off, and that's how they can behave really differently. So I'm collaborating with people actually at the Fred Hutch Cancer Center, which is in Seattle, to study a project that I did not talk about at all, but basically how tumors evolve over time and how the environment affects how a tumor evolves, and will you get a different set of mutations if it's in this kind of environment versus another? And epigenetics plays a huge role in that, rather than just mutations by turning on and off genes. And I'm not an expert in that field, but we have collaborators that are thinking about that angle.
And a research on zapping up the immune system and engineering it.
Yes. Karen Martin is a brand new faculty member that we just hired into Tufts Biomedical Engineering and started in January, and we hired her for exactly that reason. She is an immunoengineer, right? She did her PhD at Georgia Tech in the top BME department in the US. Then she did a postdoc in Norway at an immunology hospital and research center to get this very specific type of skills to study at these, from engineering and immunology. And so she is trying to rewire natural killer cells. These are a subset of T cells that you might have heard about that are very good at killing things, natural killer cells, but they're short-lived. Okay, they don't stay in the body active very long. So she's trying to basically turn them on for longer and seek receptors on cancer cells to kill them. Yeah. It's fascinating. She's
Super.
brilliant.
Thought I saw one other hand. No, over there. Oh, oh, no. Okay. All right. Yeah. Well, with that, I would just like to thank
Thanks for the
Shelley.
conversation. Happy to stay and talk.
It's exciting to hear all of this going on at Tufts. And I hope that you enjoy the rest of your time here and your class dinners tonight. And yeah, just go out there and spread the word. All right. Thank you.